people that I don't know. So welcome to those of you who have never visited us before. We are lucky to host Dr. Soumya Banerjee for 3 weeks at NICTA. Soumya recently got a PhD in computer science from the University of New Mexico. And during this time, he also spent a lot of time working at Los Alamos National Laboratory. He worked with very prestigious and famous people in both places. This is going to be the first one of the 2 seminars that Soumya will be delivering. The next one is in this very same room, I think it's next Friday at 2 p.m. And in this first lecture, Soumya is going to introduce us to the fascinating world of computational immunology. It's also a very complex world because, as I have learned this week, it requires knowing biology, physics, robotics, machine learning. So thank you for visiting us, Soumya. Whenever you want to start, go for it. Thank you.
Well, just wait for So thanks a lot for coming to my talk. And first of all, thanks a lot to Pascal and Manuel for having me over here. And I have had a great 1.5 weeks and still more time to go. It's been fun.
More crazy weather to go.
Yeah, more crazy weather, which affects the immune system. And I'll tell you what effect the weather has on the immune system. This will all make sense after I'm done with this. So the last 5 years of my life have been devoted to looking at scaling in the immune system. And first of all, I just want to thank my collaborators. I mean, this always gets pushed till the end. My advisor, Melanie Moses, at the University of New Mexico, and the amazing Santa Fe Institute. Alan Perelson at Los Alamos National Labs, also Santa Fe Institute. Our department chair, Stephanie Forrest, UNM, again Santa Fe Institute. Terran Lane, our machine learning expert, no wonder he's at Google now. And Frederick Koster, he is our experimentalist collaborator. He taught me almost whatever I now know about immunology. And I'm really thankful for the opportunity for, having been able to work with these fantastic people. They have taught me most of what I know, either about computer science or biology. So just a heads up on what my research interests are. It's in this new and emerging field called computational immunology. Very simply, it involves a two-way communication between computer science and immunology. So what I do is apply techniques from computer science to study data sets from immunology, looking at sparse time series data. And this is what this talk is going to be focused on. And I'm also interested in taking inspiration from the immune system and seeing if we can build more efficient human-engineered distributed systems or human-engineered systems in general. So this talk is just going to focus on this first part here, applying some heavy mathematical machinery from computer science to analyze these data sets and see if we can make some quantitative statements about the immune system. So this work is also motivated by understanding zoonotic diseases, which are diseases that jump from species to species. A very relevant problem, causing almost a few billion human cases annually, and things like avian influenza, which originate in birds, and mad cow disease in cattle. It's really not known how immune response rates and rates of pathogen replication vary with body sets. So we know that it's a very pertinent problem, and we know that there are differences, but there's not a single theory which accounts for all these differences across mass and phylogeny. So what I'm going to show you here are mathematical models which we use to investigate how body size determines rates of pathogen replication, rates of immune response, and then we will try to see if, using these models, we can predict how competent some host, some infected animal is for actually passing on the virus in the wild. And again, I'll give a brief introduction to what scaling theory is. It originated at the Santa Fe Institute, and I'll give you a brief introduction to that. But it's really, again, unknown how scaling affects the immune system, how body mass and these other variables affect it. Previous scaling work at the Santa Fe Institute and at the Los Alamos National Labs looking at biological systems have focused on directly observable traits like how do heart rates or rates of respiration change with body size. The immune system is a complex system. I hope to convince you of that. It's dynamic and very short time scales, and there is this arms race between the host immune system and the pathogen with nonlinear interactions. And that really means our approach is to get hold of in vivo experimental data, which means these are experimentalists who have taken an animal, injected it with some dose of virus, and then they monitor what that virus does over time. As opposed to in vitro, experimentalists taking cells in a petri dish and looking at that. I'll tell you why that approach does not work for us, and why we need to use these mathematical models to actually infer virus production. So our approach is, and so our approach is to fit nonlinear mathematical models. And do I need to do something about this?
Too late.
So you can image the immune system in vivo. You can look at what happens, but those involve techniques. Those are different techniques. Our approaches are to fit these nonlinear mathematical models to in vivo experimental data, and we do that across a wide range of species. And for reasons that I will explain, it's really not possible in our case for the questions that we are interested in to actually pull these cells out and look how they behave in vitro. So again, our contributions are we have developed a technique to apply these hierarchical Bayesian, these machine learning or statistical, if you will, techniques to infer viral production and immune response rates across a large number of species. And we test how rates of pathogen replication and immune response vary with species body size. So here's an outline of my talk. I'll introduce you to West Nile virus, which also happens to be a pertinent problem in Australia. I'll give you a brief one-on-one on immunology. Then I will tell you about past related projects and how there is an underlying theme in my research. I'll tell you about these differential equation models that I have for viral growth. And then I will take you into the magical world of machine learning and what we're doing with that. There is a role of phylogeny also in all of this, and that will play into these machine learning techniques. And then we will produce predicted scaling relations for pathogen replication and immune system response. I'll apply it to prediction of competency of a host, that how competent a host is for actually passing on infection. And then I will conclude with applications and summary. So here's a 101 on West Nile virus. It's a flavivirus, which means it belongs to the same family as something like hepatitis C. It's spread by mosquito vectors, and it infects a huge number of species. So almost hundreds of vertebrate species, which makes it a great model pathogen for us because it allows us to test how rates of West Nile virus replication change with body size. So you can look at rates of replication in birds, you can look at that in horses, and in other species. And this map that I have for the US shows that it first hit the US around 2000, and then pretty much within a few years it spread throughout the US very, very rapidly. So it's a very relevant pathogen and even a big problem in Australia. This slide is meant to intimidate, not to inform. This is a cartoonish depiction of the immune system. And immunologists would take offense at it, and justifiably so, because this is very, very simplified. What's going on here? This I just adapted from a textbook on the immune system. So you have some virus which comes in. One type of immune system cell stimulates another immune system cell to do something. It does so by secreting chemicals called cytokines, which act on other immune system cells to change their types, direct it to do something, and then it acts on other immune system cells. And this whole picture is different depending on what kind of infection you have. It's going to be different for influenza, it's going to be different for West Nile virus. The point that I want to get across is there are millions of immune system cells, hundreds of different cell types, all coordinating to achieve a single outcome, that's a speedy elimination of an infection. And so since I'm a computer scientist, this is a great system to study. It is a complex system with no centralized control. And that's what makes it such a great system to study. This is a cartoonish depiction. Let's move on to an even more cartoonish depiction. So this is to give you a feel of what this arms race is between the host immune system and the pathogen. So I have here a virus which has been injected, say, by a mosquito that infects a normal cell, which then becomes infected and secretes or churns out more virus particles. So you have this feedback loop which ensures that you have an exponential replication of virus over time. So initially you expect the virus to sort of outpace any kind of host defenses that there might be, except that there is this first arm of the immune system, this macrophage cell, which literally means big eater, which literally gobbles up anything which it suspects is foreign and suspicious. So there is this first response by this immune system trying to gobble up virus particles, but clearly this virus has a head start. And so what needs to happen is, after some time, the immune system makes a tailor-made weapon. So in this case, depicted here are these immune system cells, which after some time secrete chemicals called antibodies, which they knock out virus. And for reasons that I will get into later, this antibody response is really critical. I mean, if our models have to be realistic, they really have to capture this part. It's been shown experimentally that this antibody response is critical for ensuring survival of the host. So there is this arms race between the immune system and the pathogen, and that's what our models try to capture. Okay, now back to scaling theory. So this is a theory which first originated at Santa Fe Institute, looking at biological systems, and these are centralized biological systems like the heart. And that's what I mean by this cardiovascular network. So the network looks fractal. There is a huge pipe in the beginning, and then it branches out, and this is essentially a delivery system. It delivers blood, which carries nutrients, and what happens is the topology of this cardiovascular network constrains energy delivery. So there's a centralized heart in, say, a mouse. It has to pump blood over smaller distances, but an elephant also has a single heart, and that has to work even harder to pump blood over larger distances. So you are investing more and more into this network which sends energy, and what you get is what's called diminishing returns. The net output of this, and it's been empirically observed and theoretically predicted, is that cellular metabolic rates decline with body size. So if you take a single cell in a mouse and look at its cellular metabolic rate, the rate at which it's replicating, the rate at which it's taking in energy, that rate is much higher than a cell in, say, an elephant. And this has been observed from, say, ants to whales. I mean, it's a huge, this is a very, very significant quantitative law in biology. And what this means is, this has, naturally sort of implications for how diseases spread. So suppose you infect a mouse with a virus. What that virus does is it gets into a cell and uses that cellular machinery to actually replicate itself. In a mouse, you expect the virus to actually replicate faster since it has more energy available to it. In an elephant, you have lesser energy in a single cell, and you expect that cell to churn out virus at a much lesser rate. It could have implications for the immune system also. It could mean that immune system cells in a mouse are searching more faster and replicating more faster. So it affects this theory, which is really pervasive in biology, is expected to affect the outcome of that arms race which I mentioned in ways that have never been investigated before. Yes, sir?
Does that mean you would expect it to be different for bacteria, which have their own?
You do expect it to be different because bacteria are, naturally not using cells, but they are, ultimately consuming energy. And if that energy is, so if you assume that there is lesser energy available in the bloodstream of larger animals, that could constrain it. But bacteria are, you are right, and bacteria are a very, very different case. We actually don't know what that happens. In this case, we're just looking at viruses, but I think it's fair to say it's not known how this theory affects bacterial infections.
Would you have the same races in bacteria between the bacteria and the immune system as well? So.
Yes, I think what happens is there is some evidence to show that the energy available in a bloodstream, irrespective of body size, is the same. There's some evidence showing that. So you might expect to see different scaling relationships with bacterial infection. So suppose there's some bacterial infection in blood. It may have access to similar amounts of energy in mice and in elephants.
Yeah, actually, there is also a relationship with the energy that you put into creating offspring, right? So if offspring is actually cheap, you might not care at all about the immune system. So it could actually not be so efficient.
Right, absolutely.
So
Yes.
just a couple of things. Another scaling issue is the surface area to volume ratio. And
Yes.
if an elephant's cells was active as a mouse cells, it would explode fairly soon because of breach boiling, I presume. But the other issue is presumably there's quite a difference in the metabolic rate between different cells, and there will be some fairly non-even distribution. And I wonder how that affects.
Definitely. There are. So, for example, liver cells have a very, very high metabolism. So there are. This is to say that this is a very, very general theory, but there are differences amongst cell types. Even heart cells have a very high metabolic rate. Definitely, there are those things. And so, I guess this is my end point, one argument that the metabolic rate of a cell dictates pace of biological processes and could affect the outcome of this arms race. And we are the first to actually use quantitative models coupled with experimental data to see what the outcome is. That's one way that the immune system is constrained by body size. There is sort of another problem. So I'm again simplifying this considerably, but suppose you get an infection. Think of it as some virus particle which gets injected into you. Think of that virus as a lock. And then, actually the immune system has a key which is specific to that lock. So, again, a very, very simplified description is for any infection that you can get, there is a cell in your immune system specific to that. But naturally, these have to be very rare, and they are very rare. Literally 1 in a million immune system cells which are specific to, say, influenza. And these immune system cells have to find that virus particle. And finding that, doing that physical search, is much easier in mice as opposed to, say, an elephant. So in elephant, there's this huge physical space over which these very rare immune system cells have to recognize that pathogen. So that complicates this search process.
Doesn't the elephant have more immune system cells than
It does.
So
Yes.
What's relevant is the concentration.
Yes, that's the concentration. What's called like a protecton. How much concentration of these cells do you need per milliliter? So definitely, this it's not known exactly how many more of these rare immune system cells that this elephant has. It's probably fair to assume that it has linearly more of those in terms of body size. So there is a search problem, and then there is a problem of response. So once these immune system cells find that virus particle, it's a single immune system cell, what can it do? It actually starts making copies of itself. So it starts replicating, goes through a process of what's called clonal amplification, builds copies of itself, and starts secreting that antibody, those chemicals which I mentioned. But there's a problem because antibody gets diluted in blood. And a A larger animal like an elephant has more blood. So there is more of this dilution effect in larger animals. And the net effect is that in order to get to a critical density of antibodies in blood, larger animals have to secrete more absolute quantities of antibody. So first of all, there's a surge problem, and then there's a response problem. The response has to be proportionate to the size of that. And I'm sort of giving the story away, but this property of the immune system, which is very, very sort of attractive for us, is it seems to be able to do the surge and response in time which is nearly invariant with the size of the organism. So if you look at infections in, say, mice, and if you look at infections in horses, which is 300 kilograms, in mice are like 30 grams, immense body size differences. The immune system, at least for West Nile virus, is able to surge and respond within 2 to 4 days. So it seems to have this scale invariant property, which is very, very interesting since I'm a computer scientist, I'm really interested in designing systems which scale very, very efficiently. That really made us think, why should that be so? So this is a topic of next week's talk, but I'm giving the story away a little bit. This has to do with how these anatomical structures called lymph nodes, so they are under your armpit, under your neck. These are tiny structures which are distributed almost evenly throughout your body. And we suggested that a very particular architecture of how these should be organized, how these lymph nodes should be organized. They have to balance local pathogen detection and balance it with global antibody production. It leads to a very sort of interesting physical architecture, which I will tell you next week. But that's one part of the story. That's how the immune system can mount, search, and respond to infections in time invariant of body size. That's the physical architecture. Then there is also signaling between immune system cells. So this is topic for next week, but we looked at influenza virus infections, and we found that initially influenza causes a small infection of the area of 1 square millimeter, and a human lung has the area of a tennis court. So the question that we had was, how is the immune system able to find that tiny spot of infection and send immune system cells there? And it has to do with signaling. That small region actually sends out signals which attracts immune system cells, which in turn speeds up this immune response. And we suggested using these mathematical models that speed up due to these signaling is actually higher in larger animals. But more on that next week. Now, of course, as I said, since I'm a computer scientist, I had to draw inspiration from these architectures. And what we suggested is that you can use similar architectures for human-engineered distributed systems, where the search is not for pathogens, but the search is for some amount of rare information. And we suggested that 2 distributed systems, we have a multi-robo control system here and a peer-to-peer system, could actually benefit from a similar architecture. Again, subject for next week. But for today, I really want to focus on the modeling challenges involved in actually showing and making a quantitative statement about the immune system. And this will actually involve throwing an immense amount of mathematical machinery at this problem, because there's problems with sparse and noisy data. There's a lot of uncertainty going into our models. So whatever I'm going to talk about today can be summed up as inferring these biologically relevant parameters from sparse and noisy time series data. While there's a lot of uncertainty going in. And there are 2 tricks I'll talk about. Let me just take a swig of water. But before I get to that, let me tell you more about my mathematical models. So this is again that same cartoonish depiction of the immune system, overlaid with some rates and numbers. So this infected cell is producing virus at some rate p per unit time. And this virus has some infectivity beta with which it infects normal cells. And then this arm of the immune system, these B cells, secrete antibody which then knock out virus at some rate. So it's just that arms race between the immune system and the pathogen overlaid with some numbers. And so my differential equation models just look at these rates and numbers. Specifically, I'm assuming that this picture is true in a 30-gram sparrow and in a 3-kilogram goose. So there's almost 2 orders of magnitude variation in body size there. And all I'm going to tell you is how these parameters vary with body size. And so there are 2 sort of mathematical models which describe the data very well. These are essentially SIR type models and adapted for use within the host. So this first model is called a target cell limited model. And so what target cells are essentially, this capital T are target cells. These are cells which are susceptible, which can be infected. And capital V is the number of virus that you have in the system at some time point. And these are well mixed differential equation models. So one analogy that I use frequently is you have a box full of these target cells, these T's, and these virus particles, V's. Give it a good shake, make it well mixed, and the number of interactions that you get is proportional to T times V. So those number of interactions, so an interaction between a target cell and a virus leads to an infection. And so you see a minus beta TV, beta is a constant proportionality. So you see an exit from the T compartment into the infected cell compartment. So whatever comes out of target cells goes directly into these infected cells. So I1 are infected cells. And these infected cells, so there is some constant of proportionality there, and these infected cells are producing virus at some rate, as I showed you in that previous picture. There is a problem though, because so this model also has some of that. There's a problem though. I mean, this model, as I said before, does not have that antibody response which I told you about. So you will observe in this equation it's dV dt, all this production term, minus a constant clearance. So this is assuming that the immune system has a constant effect throughout time. And that's really not the case, since I showed you, I mean, the immune system actually makes a tailor-made weapon which does take some time, but then kicks up and gives a huge boost. So this model is not biologically realistic, but what makes it really interesting is it has fewer parameters. And so, yes.
Sorry, what's I1 and I2?
I1 are cells which are currently infected but not producing virus. So that's like another realistic thing that cells don't immediately start producing virus. They go into like a latent phase, and then after some time they start producing virus. And I2 are sort of productively infected cells. Yes.
What is the second term in the second equation, that minus k I1 is the loss of infected cells due to what?
So this is loss. So this is the rate at which these latently infected cells are actually becoming productively infected. So you can think of, so what I like to think of is 1 over k. So that's the inverse of the rate. That is the time in which that's
sort of like a conversion rate from latent
Yes.
to
latent infected to productively infected. And so there is essentially this assumes that there is a constant immune response. It's not biologically realistic, but it makes parameter identifiability much easier, because you have fewer parameters. It's a great system for studying what it's doing. And then we have our favorite model, which has all these parameters, but then it has something else. It has this antibody term in here, and this is matched to experimental data. So antibody is 0 before some time, and then it ramps up linearly with time. It's 0 really because, as I said, it's due to that dilution effect. It's not that antibody is not getting produced before this time. It just takes some time to actually reach like a critical threshold before experimentalists can even detect it in some assay. And so we have this antibody term here, And the way it's coupled into the rest of the system is with that term rho. So this represents another additional clearance from the virus compartment. Yes.
Don't you have an identifiability problem with rho and
Yes.
theta?
Yes, absolutely. We have a big identifiability problem, and that takes me to my thanks for bringing that up, because that summarizes what the problems we have. We have a model which we do not like, but has fewer parameters, makes life easier. We have a model that we do like, is biologically realistic, but has more parameters. I mean, that's life when you're modeling biological systems, as many of you would be aware. And one thing that I want to mention is that you'll observe that these are nested models, which means that given the same time series data, you will get the exact same fit to that data. So there is no way to actually discriminate between these models given a single time series data. And in the next slide, I'll tell you how we actually come up with a novel way to actually discriminate that. But let me take you through a short detour of what is involved here again. And this is where there is a dialogue between experimentalists and me, this modeler. And so it's not, so you have some time series data, which I will show in the next slide, but that's not enough to tell you, exactly everything you want to know about the system. So one thing that we have a lot of uncertainty about, so that's the first source of uncertainty, is how many of these target cells, these susceptible cells, do you have to begin with? And that's when we went into like a 6-month discussion with our experimentalist collaborators. How many cells does it infect? It's probably known in vitro, which means it's known in cell cultures, but in vivo, within an animal, it's expected to be very different. How many cell types, where does it go? And what ends up happening is, we put very generous bounds on what this parameter could be. We essentially said, okay, we have some mean estimate from literature, from talking with people, but let me just put 2 orders of magnitude variation into that, and let's see what comes out. So we were very liberal with that. Maybe we are not accounting for some cell type here, maybe we are not accounting for some other process. So let me just put very generous bounds. So that's one source of uncertainty. And I will tell you about another source as we get into our time series data. So since I work with people from machine learning, you will see that there's 5 data points in here, and people deal with hundreds of thousands of. So I don't think I have to I'm telling you that this is sparse. This is definitely sparse. Straight away, this introduces a lot of uncertainty into your parameter estimation. So let me tell you what this is. So these are experimentalists who have taken a known dose of virus and injected it into the footpad of a mouse. And then they are monitoring virus concentration in blood over time. So this is days after infection on the x-axis. On the y-axis is virus concentration in logged units in this funny unit, which is really not relevant right now, but it's logged. And straight away you will see that there is, so these red data points are experimentally measured data points. You will see that there is a linear increase on the log scale, which tells you that the virus is sort of winning this first round. But then something happens, and then it gets shut down. So I've already told you about 2 sources of uncertainty. There is something interesting about this data set. So again, sort of a recap, we have 2 models. We don't know how to actually discriminate between these. Fortunately, there is something very interesting in this data set. This happens to be what's called a knockout mice. So these are mice which have been genetically bred to actually not being able to mount an antibody response. So these are mice which actually are not capable of mounting that antibody response, which I told you is so important, which is why they die after a few days. But also means that even a constant immune response over time actually brings down the virus, which means that this is due to a process which we call target cell limited. So the virus is actually literally running out of cells to infect. And after it's literally devastated this animal, it just runs out of cells to infect and virus levels comes out. Which is sort of fortunate for us because we can now fit our target cell limited model to it. And by fitting, I mean we can estimate those betas and Ps, I'm telling you how fast is West Nile virus replicating in this mouse, we can get that. And then we have another data which is called wild type. So these are mice which have an intact immune response, and you can see the effect of that. So in blue right here, that's what this immune response does. So virus levels are much lower due to the effect of antibody. It takes some time to come in. It's probably in here by day 3 or 4. But when it comes in, it just knocks it right. But as you mentioned, I mean, this model that we like with the adaptive immune response has even more parameters than we can estimate. But that's sort of okay because we have estimated some parameters in the target cell level model, and we can carry forward these parameters into our next model. We don't really need to estimate those again. What this means is, I mean, just the underlying assumption here is that the infectivity of West Nile virus is something native to it. It really does not depend on the kind of breed of mice that you're looking at.
How do you fit the parameters?
How do I fit the parameters? So what I do is, so I told you that there's a lot of uncertainty in those parameters. So I sample at random from those intervals and do a brute force search, an absolute brute force search through this parameter regime, and I try to find if my solvers give me a solution which fits the data well defined by some threshold of difference between model predicted virus concentration and experimental data.
So you're just using least squares?
Yes, just least squares. Yes. But what I'm doing is I'm giving my differential equation solvers millions of different random combinations of these parameters sampled from those. So for something like the target cell population, I randomly sample from those 2 orders of magnitude variation and give it to my differential equation solver. And literally tell it, I mean, can you find a good estimate of this parameter? Can you find a good estimate of rho? Can you find a good estimate of beta? Some very, very simple brute force approach.
Do you normalize to when you do the squares, do you normalize it to the absolute value of the data point? Because for there are very large variation in the orders of magnitudes. And
so what I do is I use a threshold, and it's a very, very subjective threshold admittedly, but I say that only consider solutions which are within, say, 0.5 small square residuals in whatever units this is.
no, what I mean is that your data points that are high will influence your fit much more than data points that have lower values.
Are you doing least squares on the log scale or with raw data?
On the log scale. On the log scale, yes. So are you saying that there's an assumption that the parameters are log normally distributed? Is that?
Well, it means the large data points are much more influential.
Yes, that's right. So there is. Oh, I see. I mean, there's no weighted. There's no weight here. There are equal weights to each of these data points. That's what your question is.
And do you expect the noise to be symmetrically distributed?
You mean the parameters to be symmetrical?
But this model is deterministic,
Yeah.
so
Yeah.
it's not a stochastic model, right?
It is not stochastic.
Maybe it should,
you mentioned
but it's not.
Oh, but if you're doing least squares, it's like adding normal errors to the data.
Sure. I mean, let me go to the next slide. I think this will make it clear. I mean, so I do this. So is this process clear to everyone on what I'm doing? It's a novel combination of knockout experimental data, which means I can fit my target cell limited model to it, and my wild type data, which means I can fit my adaptive immune response model to it. So that's our trick 1. There's also a trick 1B. So this is what comes out. So here's a frequency histogram of one of these parameters. This parameter tells you how effective antibody is at knocking out virus. And we didn't have any experimental data, so we just said, okay, constrain it to lie between 0 to 50. So that is the amount of uncertainty that went in. And after this procedure, when we do that, we get as much uncertainty back. You know, to say that there isn't like a well-peaked distribution, there isn't a single value, there isn't like a good distribution which describes rho. In other words, there is a parameter identifiability problem. We cannot predict these individual model parameters, and this is true for other parameters also. You cannot predict individual model parameters with a lot of certainty. But something interesting happens when you look at sort of combinations of model parameters. So one thing that I want to talk about is the R naught, and I just want to see if I'm doing okay with time. 10 minutes. Okay. So one thing that I want to talk about is this within host R naught, which is this basic reproductive number. And if you think of this infection process as a critical chain reaction, you have one infected cell initially, then that produces 3 more. Each of those produces 3 more. So the R naught for this is 3. And this R naught is a nonlinear combination of all those model parameters which I told you about, betas and P's and all that. And what happens is, if you look at these biologically relevant quantities which happen to be nonlinear combinations of those model parameters, you get some certainty out. So there's a frequency histogram of the R naught, and you expect it, I mean, given all the uncertainty that you have in the model, you expect it to vary from 1 to 30. But you see that this R naught is actually almost well constrained, between 1.4 to 3.4. So you don't have any certainty in individual model parameters, but you do get some information when you look at combinations of those parameters. So that's like our other trick, and those combinations turned out to be really biologically relevant. There are other quantities which I'm not telling you about, but knowing the within host R naught tells you something about the system.
So,
Yes.
I might have missed it, but what is it that you're varying for each one of these replicates in your histogram?
So I'm varying values of P. There's variation in T naught, which is the number of target cells that you start off with. Then there is variation in gamma, which is like an immune response term. So there is a lot of uncertainty going into each of these model parameters.
So each of those things is varying in response to what? I mean, you just...
Well, what I'm doing is, so there's variation. I give that to my solvers, and then it gives me a solution back, which produces a good agreement with data. So this is, non-linear least squares. It's trying to minimize the distance between model predicted virus concentration and experimental data. So it gives me, I give it certain values of parameters, and it tells me whether those parameters produce a good prediction.
How do you select the threshold?
The select, that is absolutely subjective. I looked at like what seems like a good fit to data. If it's like too high, so that turned out to be something like 0.5 or 0.7. I don't remember the exact values, but that is a subjective threshold. That, we can definitely look at other thresholds, look at distributions around those. But that is a subjective threshold. Okay, so I just want to make sure that I run through some of my other things. That was for mice. We also have data for birds. So West Nile virus causes a lot of infection in birds. And in birds, unfortunately, you don't have these knockouts to actually rescue. It's very hard to make knockouts for birds. Fortunately, we do have some other information. There is information about, so some of these birds, these smaller birds, known as passerines, denoted by P here, are somehow more susceptible to West Nile virus infection than some other, these sort of larger birds known as non-passerines. So passerines P are more susceptible to West Nile virus infection than non-passerines NP. And you can see the joke here. Probably my computer science joke is that this proves P is not equal to NP. I wish it were that simple. But essentially, what it gives you is, what I want to do from now is, I want to encapsulate whatever information is there in this hierarchy and use that to extract some more information from this sparse sequence data. And our technique here is to use these hierarchical Bayesian models to actually encapsulate some of these differences as priors into my system. And again, what this means is, so I have all these individual birds. So I have 23 species of birds, about four birds per species. So I have about 80 to 90 birds. And so there are, for each of these birds, they have their own differential equation. They have their own data points, so it's still data driven. And then on top of this, I have my hierarchical variation model. And there is no magic here. These models are still data driven. What is happening is, I'm giving it some extra input from the top, telling it that, I expect something to be fundamentally different about some parameters in B and something different about NP. So there is some more information going into it. And as I will show you, that makes a lot of difference. That produces more accurate estimates. And these represent millions of calls to my differential equation solvers, and that will lead to some engineering problems, which I will tell you in a minute. And I'm going through fast here, but essentially these hierarchical variation models model between and within host variation, and they allow you to pool information. So you're not just fitting to one species here; you're fitting to 23 different species, 83 different birds, and maybe there is, even though there is sparse data at the level of an individual bird, maybe pooling different birds together can allow you to circumvent that and give you more information. So that's what this is. Just want to say that these are nonlinear mixed effects models, and Bayesian nonlinear mixed effects models with multiple levels of hierarchy haven't been applied to us strongly before. And as someone important said, we really need to do that. So I'll skip over this. I'll just tell you quickly about some engineering problems. So this is literally 6 to 8 months of work. This is the graduate student down in the trenches, fighting for months, not knowing what's going to happen the next day. But essentially, I had to make sort of assumptions going into this. And one big assumption is that all these differential equation parameters are log normally distributed. That's just to ensure that the math all works out okay. And then I have to derive equations at each of these levels, which means applying Bayes' rule at each of those. And then there is the engineering approach of actually getting it to work on multiple cores. So I'm using MCMC to do inference, and just getting it to run fast. So there is that engineering aspect of it. And what I'm comparing against are hierarchical models. So this is my what I call a multi-level model with a species level representation. And what I'm comparing against is an aggregated model without that species level representation. And I'm sort of running out of time here, but let me tell you that it wasn't, so we really have to do a validation to see, under what circumstances these complex multi-level models actually outperformed simpler models with fewer levels of hierarchy. And I'm just going to skip through these really fast. I use a toy model to generate data with known amounts of variation, and this plot tells you that it's expected these multi-level models with 2 or 3 levels of hierarchy are expected to make better predictions when there is more between species variation than within species. So in this plot, lower is better. Our favorite models with multiple levels of hierarchy are in red, and green is with fewer levels of hierarchy. And again, lower is better. So in this particular case, when there is more between species variation than within species, our models do better. So it also does well on our actual experimental data. It produces better predictions, and here is how much better this is. So here's a prediction produced by our multi-level model in blue, and just models with fewer levels of hierarchy do much worse than this.
And the black points experimental data are
Yes.
similar like mouse experiments that you did before? Or
These are
birds?
similar. These are now in birds.
Yep.
Yeah, but yeah, similar. The only difference is that these are now sampled daily instead of on alternate days. Yes.
Is each data point a different animal or the same animal?
This is the same animal. So this is probably an owl. So this is just one individual belonging to that.
So that
species.
takes blood every now and then?
Yes. And again, this is another frequency histogram of that basic reproductive number, which I told you about. This is now on a log scale. And straight away you can see, so NP is in blue and P is in red here. And so you can see that there's a lot of overlap. But if you look at the peaks of this distribution, so right here, so the peak for P is much, much higher than the peak for NP. So it's telling you that the basic reproductive number, that the length of that branching process is much higher for these passerine species than these non-passerines. So there is.
Why do you have this bimodality?
So in the green curve.
Yeah.
So in the green curve, I think
in the red one, so it's a little bit, right?
there's a little bit, but there's more in the green one because these are an even smaller subset of passerines. So these are corvid species like crows, and I think we didn't have much data to actually make. If you have less data, this is what tends to happen. Having more data sort of evens out.
So you think it's just a detail and actually as a mode and
Yeah.
okay.
Okay, I'll skip over this. It just tells you that our aggregated models do even worse here. So here's what we're really interested in. So scaling of P, which is the production rate. So at what rate are these infected cells producing virus particles? So we looked at, so there is a measure for P for each of these species. We have a mass associated with each of these species, and I'm doing a linear regression with that. And this is showing you that there is a statistically significant relationship, declining relationship with the cells. So
What is the second code? Is 1.10 or 1.17? Do you have it?
what's
In
the
slope.
second one? You mean?
Yeah. What is the second digit after the 1? The slope.
You mean the slope?
Yeah.
I think it's 1.17.
Yeah, so it's like cities, right?
That's an interesting point.
Yeah.
That's
It's like road
Yeah.
networks
Yeah.
and electricity, right?
Yeah, that's an interesting point. So the rate at which West Nile virus is produced by infected cells is slower in larger animals. That's a simple message here. And but if you look at rates of adaptive immune response, and in this case we looked at antibody response, those do not have a significant relationship with body size. With the caveat that within passerines, so passerines is in black, there is a significant relationship. So again, overall, if you look at P and NP combined, there's no significant relationship with body size, with these rates of adaptive immune response. Similarly, the time taken by the adaptive immune response to actually start secreting antibody and get antibody into a critical level, that also does not have a significant relationship with body size. That's also gets you into that nearly scale invariant idea. I'm running out of time here, so I won't tell you how we can come up with predictions for what happened to my slides. Okay, so summary. So these results are consistent with sort of scaling theory hypotheses that rates of production of virus should decline with body size. However, rates and times of adaptive antibody response do not decline with body size. There is an influence of phylogeny, which is what our hierarchical Bayesian models capture. And together, mass and phylogeny give us mechanistic insights into how West Nile virus is actually spread. Larger non-passerines are actually sort of dead-end hosts as far as the pathogen is concerned. And so one thing that I sort of wanted to get, everyone to get out, and this will become more apparent in my talk next week, is thinking about the immune system as a computational system. It has implemented this remarkably efficient distributed search for pathogens, and immune response is nearly as fast in small and large systems. And if you think of the physical architecture, how these lymph nodes are organized as the hardware, and how these immune system cells actually communicate with each other as a software, that that as an inspiration can help you build more efficient distributed systems. More on that later. I have to sort of sell myself. Unfortunately, I have to be the salesman. I have to sell myself, but just one slide. So this work is, I mean, could be even more broadly sort of relevant. I mean, inferring biologically relevant parameters from sparse and noisy time series data. These are two techniques that we have looked at. If you have any kind of hierarchical information, you can exploit that using hierarchical Bayesian models. And I have experience modeling using continuous differential equation models, and I have also spent a lot of time building these discrete, spatially explicit agent-based models. And then I have also an extensive programming experience on multiple codes, and I'm within the top 500 MATLAB code contributors in an open source. The last thing is I have a lot of experience working with experimentalists, and that's what really made it worthwhile. I mean, that's one part of my job that I really enjoy, having a two-way communication with experimentalists, how much detail to actually incorporate, what to leave out, and they keep on reminding me that the immune system is vastly more complex than we can even begin to imagine. Thanks a lot.
Questions?
Yes.
The West virus, does it adapt in each of the different hosts?
It is, yes, there's some evidence to suggest that it is. I mean, it's probably very well adapted. So this 1999 strain that we looked at, that's very well adapted to replicate within a particular subset of these pastorals known as corvids. So huge amount of deaths in crows, which are these corvid species. And it's suspected that there is some mutation in the virus which has made it very infective.
Yeah, I'm just thinking that from a long-term evolutionary perspective, okay, if it's got a very different phenotype in crows, that suggests that it's not. I mean, you can still go between them, but you would think that how an animal's immune system works and how fast it can clear stuff depends on its lifespan and what maximises its fitness for reproductive purposes.
Yes.
So there is some sort of what you're observing is post-evolution.
Yes, absolutely. I mean, what we are observing is post-evolution, and you are very right. I mean, there is some work suggesting that lifespans do have an effect. So most viruses come in through the respiratory tract, and rates of respiration. So if lifetimes are longer, you have a higher probability of getting infected with more viruses and more virulent ones. So there could be trade-offs.
Bigger animals tend to live longer. I mean, birds would be an exception.
Yes. Yeah. Bigger animals, I mean, they also tend to live longer because of that metabolic scaling theory which I told you. So it turns out that the number of heart rates is conserved. So a mouse has as many heart rates as we do. It just consumes its heart rate, just beats much faster and lives much, for a shorter time span than we do. So we have a lower pulse rate, but we sort of stretch it out over a larger lifespan. So you would get that from that also. But yes, you're right in suggesting that, I mean, what we are observing is post-evolution.
Yes, so how fast you can clear something, I mean, you get your immune system to work well enough that you can do that in a way which is worthwhile. Spending too much resources doing that.
Right.
I have two questions that are actually one and are related to the other one. So this scaling variance in clearing time, do you see as the result of coevolution between the immune system? So because, scaling variance in heart rates, that's not coevolution of anything, right?
No, that's not. It's a static system.
Exactly.
There's
But this one might seem that it is.
Yeah.
So what is your personal intuition? Because you didn't talk about this in the analysis.
So
what is your personal intuition about
my intuition is, and again I should strike a cautious note because we have only looked at West Nile virus. And the immune system is not optimized for clearing just West Nile virus. I mean, it has to probably consider a whole universe of pathogens. But I think there are some very interesting co-evolutionary things here. I mean, why doesn't the immune system, say, go down? Isn't there a different scaling from what we observe? And as we were talking about, I think there are there are sort of other quadrants, other cases which have been ruled out by these co-evolutionary arms races. It's I think reasonable to suppose that the immune system response rates actually increasing with body cells. Maybe that turned out to be very, very costly sort of from an energetic standpoint, and maybe you didn't even need to do that. And those probably got weaned out by natural selection. It is possible.
The second question is, why isn't there experimental evidence or an attempt to study the coevolution between the immune system and the pathogens?
So one thing.
What is the experimental limitation there?
So the first limitation is, so if you take, so suppose there is a mouse and there's a cell within it which is infected. If you take that cell out, that is out of that cardiovascular network, and immediately what happens is its metabolic rate settles down to a baseline metabolic rate. Same for a cell in a network. So you have to do these in vivo experiments. Now there are imaging techniques to actually image within sort of in vivo and look at a lymph node in vivo and look at those. I think one sort of limitation is that you have to inject different chemicals into that local area, and that could change metabolic rates and things like that. It definitely could be done, can be done, but it's not been done. And our approach has been to use these models to actually do that. But I think there are some experimental hurdles to actually doing that. But it is possible. Yes.
My central question is, back on, you had a distribution of rho.
Mm-hmm.
Now,
let me go back to that.
just get back there. That slide and the next slide. I just want to concentrate on that one.
Okay.
From a statistical point of view, the distribution of rho looked to me to be made up of a mixture of 3 distributions.
Okay.
But
Yeah.
you've got an excess of zeros,
Mm-hmm.
and then you've got a middle section,
Yeah.
and then you've got, I don't know if the 50 is just an upper bound,
Yeah.
there's everything above that.
Mm-hmm.
So I'd like to just ignore the upper bound part.
Okay.
So we can have a mixture of 2 things. One is a mixture which is a lot of essentially excess zeros.
Mm-hmm.
Now that occurs a lot in statistics where you when you try to scale things. The simplest example is the Poisson distribution.
Okay.
Everything's got to be, so by the way, everything's positive.
Okay.
It's either positive or 0.
Mm-hmm.
Right? And you're thinking of the pathogens. I was thinking of this whole problem from a statistical point of view. The pathogens are rare,
Right.
right? And therefore the concentration of them is close to 0.
Mm-hmm.
And so you've got a whole lot of, in the body,
Mm-hmm.
no matter what size, a whole lot of non-pathogens.
Mm-hmm.
Right? So there's a whole lot of zeros and a few ones.
Uh-huh.
And this is the spread of the ones, and so I was thinking, I don't know if I'm at all relevant, there's something going on, if you like, with the response rate which is affected by the zeros,
Mm-hmm.
and there's another part which is actually affected by the actual pathogens themselves.
Rather than themselves.
And so even in that diagram,
Mhm.
I see that as a mixture of at least 2.
At least 2 different
And
processes.
when you get to your next slide,
Mm-hmm.
we just have your next slide on there, this one,
Mm-hmm.
by using this ratio here, you've got rid, in a sense, of all the 0 cases.
Yes, because this.
Because you've done the scaling and all
Right.
the mixing,
Right.
right? And so that's tidied the whole appearance.
That's a good point. Another thing that we do observe is, there are correlations between these parameters. So it's not like I have 2 orders of magnitude variation in P, I have 2 orders of magnitude in delta, so I expect P over delta to have some maybe 2 orders of magnitude variation. These correlations constrain the possible space of combinations which are feasible. So our data fitting, which minimizes the distance between model prediction and experimental data, actually weans out all those other infeasible combinations. I think that's what happens, and at the end, what you end up with is a very narrow constraint space.
So, just One more minute, then I'll be quiet. I thought the zero cases were where you're essentially searching for pathogens and basically you don't find any.
Right, or I mean since that's a rate of response, I mean this could mean that you don't need antibody in some of these solutions. So maybe this represents like a target cell limited ratio.
Yeah.
You don't need that much
And
antibody.
the other part essentially is when you do find a pathogen, you then probably just search locally,
Right.
so you get a faster rate.
Mhm.
But instead of going elsewhere,
Mhm.
if you found something,
Mhm.
look in that neighbourhood.
Right. So you're saying that there could be spatial effects about how the pathogen is actually.
When we have, often on humans, we observe sores, don't we?
Mhm. Yeah,
Right? Where there's a concentration,
it is localized.
Localized.
Yes.
and I expect that
Yeah.
the sense of the virus is multiplying more rapidly in that
Mhm.
locality.
Right.
And
That's
I don't
an
know if that's
interesting
if you
suggestion.
do your models,
Yeah.
but that was, I was just thinking from a statistical, lots of no response.
Mhm.
But where you do get a response, it tends to be localised
Mhm.
if you're going to be efficient.
Right.
So,
That's
be quiet.
that's a very that's a very good. There could be information here that could be sort of informative. There definitely could be something in this distribution. But we just found it interesting that we have this sort of an identifiability problem, that this parameter is still able to vary over this large range. But you're absolutely right. We should spend some time looking at maybe these boundary conditions, and why do you get high values over there and high values near to the zero.
Can I just ask, are you using exactly the model you showed us a few slides earlier?
Mhm.
Yeah.
Yeah.
Because if you could go back to that.
To the model slide.
Yeah.
Okay. Yeah.
So you've got the rho and the eta.
Mm-hmm.
So when you do rho times 18,
Mm-hmm.
you get naught, and then you get rho eta times t minus. Yeah, so
So
if
you
you've done
get zero.
Yeah, but it's the rho eta. So those two only ever appear as a product.
Right.
So if you've done it exactly that way,
Mm-hmm.
you can never identify.
That's correct. We
So you
cannot.
should just put eta equal to 1.
Actually, that's a very good suggestion. You're right that we can only identify rho times eta. Fortunately, we have actually experimental data to tell us what eta is.
Oh, okay. So,
So
you-
we
you've got
fixed it at
from
that
somewhere
value.
else.
But you're right in suggesting, in this model, we can only identify rho times eta. Fortunately, we just happen to know what eta is
Okay.
from experimental data, so we just fixed it to that. But
I had another question
Yes.
about your hierarchical model.
Mhm.
Did you try specifically modelling the linear or the effect, of mass as an extra level in the hierarchical model?
Yes, we did.
You
So
did.
that's what we did in this validation stage. So I did not get time to go into that. But in this validation phase, what we did was we looked at a very simple toy model to actually generate data, and the model is dVdt is equal to A times V. It doesn't get any simpler than that. And then we incorporated mass as an additional variable into our hierarchical equation models. Yes, and that actually gave us a lot of power, because that's what our other model doesn't have. It doesn't have that mass level or that species level representation. So you get some more information out of that. But we did look at having mass in here.
Guys, were you using informative or non-informative?
So we have somewhat uninformative priors, and I say somewhat because some of these parameters. So for it will probably make sense if I go into the real model. But for some of these parameters, we have actually more information than others. So we know, what kind of dose these mosquitoes actually inject. So we have more information about, say, V naught, which is the initial number of injected virions or virus particles, than some of the others. So we had sort of very informative priors for that, and for some of the other ones, we had to choose very sort of liberal bounds again, very wide distributions. Yes, uninformative priors.
So I know that some colleagues here have to go to a meeting, so if you have more questions, Soumya is going to be here for almost 2 more weeks.
Yeah.
So maybe you can take them offline, so let's
Yeah.
send Soumya again once more.